Hi, I'm Brian Canton, grower specialist for Berger peat moss. Today we are going to talk about creating the ideal root environment. Here at Berger, we go to great lengths to understand the interaction between the physical, chemical, and biological properties of a rooting environment. Without going into much detail, the physical properties can be defined as how the components nest together to produce macropores for drainage and oxygen content, and micropores for water retention. Chemical properties are represented by the EC and pH values created by adding lime and fertilizer to the peat-like mixes. Later on, we will expand on the chemical properties when we visit the tech lab. Biological properties, simply stated, are the naturally occurring beneficial microbes found in good quality peat. One of the main criteria in designing a mix is how fast the mix will drain of excess water and how quickly oxygen will return to the mix. Oxygen is a precursor to many plant and root activities. As a plant physiologist, oxygen sets precedent over nutrient availability. Creating an oxygen-friendly mix begins with the proper selection of particle size. Each component is sized to complement each other in creating a root environment with ideal drainage, water retention, and aeration. As part of quality control, we utilize the expertise of Berger's production and technical lab to monitor particle size. Creating a tight mix where oxygen is limited for prolonged periods of time jeopardizes the production of root hairs. Root hairs are elongated cells found along the main root. Root hairs are also the main receptor for nutrient elements. The effort that goes into producing a root-friendly mix can be jeopardized if the mix is improperly handled at the greenhouse site. I will not elaborate on this subject since we have produced a DVD titled Best Practices in Media Handling. Our R&D department is constantly trialing mixes to assess root development under different fertilizer and water regimens. Quite often, the above portion of the plant does not express any visual symptoms until the root damage has occurred under stressful conditions. Compared to a healthy root system, wet and high salt conditions can jeopardize the root development. Let me show you what to look for under high salt conditions in a root system. As you can see, the roots are starting to brown off. Unfortunately, this root system was not observed on a daily basis, and the salts started to build up frequently. The symptoms are not showing yet on the top of the portion of the plant. However, in 2 or 3 weeks, as the salts get higher, we would start to actually see burn on the leaves. And now let's look at a root condition stressed by overwatering. First of all, you can see that the size of the top of the plant has been greatly reduced due to the lack of water uptake. The actual root production is very minimal, the main reason being the lack of oxygen. As I had indicated before, oxygen is the precursor to root hair development. And if you were to look closely at the damaged roots, there is quite an absence of root hairs in this wet pot. This root system represents an ideal Berger growing mix. The interaction between particle, chemical, and biological properties have defined and produced a perfect root system. We will now proceed to Berger's technical lab, where we will walk you through our daily quality control practices.
Hi, I'm Pierre-Marc de Champlain, project coordinator here at Berger Peat Moss, and I will walk you through our tech lab to show you what we do for quality control. Initially, we receive samples from the production plant which are systematically taken off the mix line and are labeled with a barcode in order to properly identify them. Now, with the sample, I will take you through the complete SME analysis. A fraction of the mix is placed inside a beaker, which is then incubated with distilled water. We let the mix rest for 2 hours. It's been 2 hours. I will take the sample to Robin so he can take the pH. The next step is to extract the liquid from the mixture in order to verify the chemical characteristics and make sure they fall within our specifications. The liquid extract is then processed in the FIA, an automated equipment that allows us to determine the EC as well as the nitrate and ammonium content of the sample. The extract is also processed in the ICP, which allows us to measure the micro and macro elements. With the LIMS, the results are automatically transferred into our system and are thoroughly inspected by a qualified chemist who then produces and approves the final reports. A complete SME analysis only takes 48 hours from the moment we receive the sample.
Sample kits are available for clients who require a tissue, water, or substrate analysis.
At Berger, we also have a microbiology lab, which allows us to measure and identify the microbial populations that are present in the samples we receive.
In conclusion, it is important to realize that an extensive root system is a prerequisite to the overall health and quality of a plant. I would like to thank you for your attention, and keep in mind that here at Berger, we are truly the root environment experts.